Okay, so I'll just start. So hi, so for those who don't know me, so I'm Steven Gazal. So I'm a new assistant professor at the Center of Genetic Epidemiology with an affiliation to the Biostat department. And so today I'm going to present you my work that it's hopefully like ready to be submitted. And which is called like combining SNP-to-gene linking strategies to pinpoint disease genes and assess disease omnigenicity. So I've already done a talk, like I think 6 months ago, starting to introduce the methods combining SNP-to-genes. And so today I will recap this a little bit, but I will more talk about the applications. So I've planned to talk few about methods and a lot about the application. But if you have some questions at any time, I don't know, just stop me and I'll be happy to take some questions. Okay. Okay, so this is like a classical first slide for a talk talking about method on GWAS data. So this graph is showing a Manhattan plot like of a recent GWAS with nearly 1 million individuals. So for those who don't know what is GWAS and a Manhattan plot, so this represents the genome, and each point represents a polymorphism, a common polymorphism. And the y-axis represents the strength of the association with a phenotype. And so here the phenotype is a blood pressure trait. And so what we can see from this GWAS, and this is like a general pattern we can observe in a lot of GWAS, is that it's highly polygenic, which means that there's a lot of variants associated to the traits. And so this could be a great news, like just to say, okay, now we have data sets that are so powerful that we can highlight hundreds of variants, and so we can learn a lot about the biology of the trait. But what happened is that what it turned out is that all these associated variants tend to be mostly non-coding variants, which are very hard to interpret. So really what has been like the main challenge for this what I call post-GWAS era, which is like trying to interpret like all these GWAS hits, is to link these regulatory causal variants to their target genes. And so this is a tough question. I think it's not straightforward because the target gene of a regulatory variant is not necessarily the closest one, as it's illustrated on this graph, where let's say you have a red mutation that causes a disease and you want to know which gene this mutation impacts. Then it's totally like it's likely that the targeted gene is not gene A, which is the closest one, but is gene D, because chromosomes, I don't know, are like packed into cells, and it can turn out that these variant is an enhancer that interact physically with the promoter of gene D, which is like the fourth closest gene. And so one extra challenge in trying to understand like this 3D architecture of the genome is that these regulatory elements are cell type specific. So for example, you can imagine that you have a gene like gene X here and two different tissues, such as, I don't know, heart and skin. Then this gene will be regulated by different enhancer in tissue A and tissue B, which means that this 3D map is not universal, and it's and this, I don't know, just means that we need to have as many tissue and cell type as possible to try to understand where are all the enhancers and how they are connected to the genes. Okay, so one solution to link these regulatory causal variants to their target gene is to use to aggregate evidence from functional data sets, right? Because I don't know, if you do a GWAS like that and you have like hundreds of variants, it's like really hard to manually validate all this signal. And so like the common solution right now is just to aggregate evidence from this functional data set. I think this has been like pretty well illustrated in this paper related to the FTO example. So the FTO example is one of the first GWAS hits fell into like the gene FTO, and this variant was associated with BMI and obesity. And so what this paper showed is that the causal variant, which is like really in the middle of the FTO genes, is actually a regulatory variant that target RX3 and RX5. And they show that first by showing that this variant is an enhancer that interact physically with the promoter of RX3 and RX5 using high-C data. And then they also show that this variant impact gene expression of RX3 and RX5 in the relevant cell types and tissue, which was here adipose tissue. So, okay, so since this paper, like a lot of strategies has been proposed in the field to link variants to genes. So I can cite promoter capture high-C, which is like a fancier version of high-C data. And you can see that it's really like an ongoing strong research direction, because there has been like these two enhancer maps that has been published like a few months ago in Nature. The first one called the EpiMap enhancer, which is basically like the continuation of ENCODE and roadmap consortium. And this new type of enhancer called the ABC enhancers. And so what is hard now, we have like all these functional data sets in hand, is just trying to understand which one we should prioritize in GWAS studies. So for example, let's say we are in a case where we have highlighted the causal variants in the FTO gene, and let's say that EpiMap targets this variant to RX5, but ABC targets this variant to CDHD9. Like we don't know which one to trust, because we don't know which one is the more informative in the context of GWAS. And so why we don't know that is actually like the resource that is needed to validate those maps is not available. So the resource, so what we need to know is like a gold standard set of causal variant and causal gene pairs. But these are not really available yet. And the reason is that we created these functional data to highlight these pairs. And I don't know, like if you scroll the literature, there are like really few examples where both the causal variant and the target gene has been functionally validated. Okay, and so because this resource do not exist here, I will use some polygenic analysis of disease heritability, which in other words mean for a given GWAS, I'm going to use the information you observed on all the SNPs, not only the ones that pass some genome-wide significant threshold. Okay, so the ultimate goal of this work is to kind of build a super SNP-to-gene or S2G map, which will aggregate functional data sets and provide functional interpretation of for every SNP of the genome. And so, for example, let's imagine that you have a SNP here, which is RSX. What I want to build is a map that scores every surrounding gene by giving some interpretation using functional data. So for example, in this case, we can say that gene A is the more likely target gene, because we found that RSNX is an EQTL impacting expression of gene A. RSX is an enhancer that is linked to gene A, and RSX is in a high-C loop, which means that it interacts physically with the promoter of gene A. What we don't want to have as information is closest gene, because it doesn't bring any interpretation. It doesn't say why RSX target gene B, right? It doesn't help to say like functionally what is the mechanism, because apparently it's not impacting expression, and apparently the enhancer is not linked to this gene. And then we can have scenarios where like multiple genes are targeted by a SNP, but with a different weight. So for example, here we'll give like a lower weight to gene C, which has only like the EQTL information. Okay, so the outline of my talk is I will really briefly introduce existing strategies linking SNP to gene. And then I will explain my framework to evaluate these strategies and my framework to combine them by keeping in mind that there is no ground truth data set, and I will have to use some trick using polygenic analysis. And at the end, I will do some application, and I will leverage this method to better understand the architecture of disease. Okay, so first let me introduce quickly what is a strategy linking SNP to gene. So basically, a strategy linking SNP to gene is just a functional data set that allows to link some SNPs, not all of them, to some genes. Okay, and so sometimes it's going to be it's going to link a SNP to a single gene, and sometimes it's going to link a SNP to multiple genes with different scores that are kind of like normalizing these 2Ds so that they can sum to 1. Okay, so I've considered a total of 50 strategies, and I've highlighted 13 of interest here. That's right, 13? No, 11. 11 of interest here. Sorry. So basically, we have what I call like the high confidence SNP-to-gene strategies, which are exon and promoters, because I don't know, it's pretty easy when you have an exon and a promoter to link this exon and this promoter to the gene, because I don't know, each exon defines the protein related to the gene. Then we have what I call like the non-functionally informed SNP-to-gene strategies, which are like what is usually used by default to link a GWAS hits to its target gene. So you can have like the gene body, if a SNP fall in a gene body or not, if a SNP fall in a gene body plus some window to kind of include distal regulatory element. You can also have, you also just have the closest TSS or the closest gene, which is often used as a proxy for the targeted gene. And then you have like all SNP-to-gene strategies, which are built from functional data sets. Oops, sorry. And so I've considered like different strategies based on gene expression. So I have like 2 fine map EQTL studies, the one from GTEC. So in GTEC, you have around 500 individuals in 50 tissues, and I'm just keeping for each SNP the higher probability to be an EQTL of a gene in a given tissue. Then you have the EQTL Gen Consortium, which is like a big consortium with around like 30,000 individuals with blood expression. And then you have this new map, which I like, which are EpiMap enhancer, which are linked to the genes by looking at correlation between enhancer activity and expression across more than 800 cell types. Then you have other strategy based on either high-C data, either on single-cell attack-seq data, and I won't spend some time to describe them. But what I want to quickly mention is these strategies differ in the number of SNPs variable to target to the genes, right? So exon, like only like 2% of the SNPs are in exons. And the second one is that these functional data sets differ also like in the number of biosamples they have. So for example, just keep in mind that these EQTL strategies use like a lot of data, like in the magnitude of like greater than 10,000 experiments. Whereas this linked enhancer, they use like really few data sets. Basically, they detect enhancer in one cell type, and they have some methods then to target them to the gene. Okay, so now we talk about the framework I've developed to evaluate and combine both strategies. Okay, so the first, so before, so the first thing we need to do when we compare things together is just to define the metric we want to maximize. And so here in this case, there are like 3 different metrics we want to maximize. So the first one is related to the fraction of causal SNPs that are annotated. We want to have strategies that annotate a lot of causal SNPs, right? And this can be estimated by something that I call the heritability coverage, and which is the fraction of heritability explained by SNPs linked to all genes, right? So if we're going back to this strategy, for example, we can say that exons will have a heritability coverage of around 10%, because we know that 10%, around 10% of the hits in GWAS are in exon. And the heritability coverage for closest TSS will be 100%, right? Because like every SNPs will have a closest TSS, so every SNPs, every causal SNPs will be linked to at least one gene. Okay, the second thing we want to know is what's the precision of the strategy. So what is the fraction of annotated causal SNPs that are linked to the correct target genes? And this is more challenging to estimate, and I will define how we did that next slide. And finally, the last thing we want to estimate is what is the fraction of disease SNP gene pairs that have been detected. And so this is similar to recall. And this recall can be estimated by just multiplying the heritability coverage and the precision together, right? So really what we need to find here is a good way to estimate the precision of a SNP-to-gene strategy. So to estimate this precision, we made an hypothesis. And so we made the hypothesis that if we are in a region like this one with several SNPs, each SNP will have different effects of some phenotypes. And so if we have some a priori on the genes, so for example, we looking at schizophrenia and we know that gene A is expressed in brain and gene B is not. So we'll say that gene A is in a critical gene set. Then our hypothesis is that a precise strategy is more likely to link the SNP with a high effect size to the gene in the critical gene set. And strategy that is less precise will link this SNP with high effect to the gene that is not in the critical gene set, right? And so when we generalize this and aggregate this signal across the whole genome, we have the intuition that the precision can be computed as a function of the heritability explained by SNPs linked to critical genes divided by the heritability explained by SNPs linked to all genes. And that ultimately, like a very precise annotation should maximize this ratio. Okay, so now we just did some normalization to convert this ratio to a precision value, which is between 0 and 1. And so to do that, we just made the assumption that like the exon SNP-to-gene strategy is perfectly precise. It has a precision of 1. And when you take that into account and you do like some, I don't know, normalization, you have some function of that can be converted into a number between 0 and 1. And this will be our precision. Okay, so now I've mentioned that this hypothesis rely on the use of critical gene sets. And so we use two types of critical gene sets, one to train our method and one to validate it. So for training, we use the same gene set for all the traits, and we kept the 10% of genes that are that have the most conserved exons and promoters. So we're just saying that, okay, like this set, like the genes for which exons and promoters are conserved across species are very important. And this is true for all the traits. And so to validate our framework, we built some trait specific gene sets. And so to do that, we run the recently developed PUPS method that prioritizes genes in disease, I don't know, by aggregating evidence from multiple gene set and pathway. I won't describe that, but it gave like pretty nice results in terms of heritability. And so, okay, so now we have our framework and we will estimate our heritability coverage, precision, and recall on 63 independent traits. And we will meta-analyze the results to have kind of robust estimations of these values. Okay, and so now we have in hand a framework to evaluate the strategy. We can build an optimization algorithm that will optimally aggregate SNP-to-gene strategy. So for example, let's say that we have some gene here, and so we can link SNPs to this gene using different approaches. So using either exons, promoters, and also like enhancer maps. And so what we want to do is just to estimate some weights for each of these SNP-to-gene strategies. And of course, like these weights will be, I don't know, will depend on like the precision and the recall of the strategy. And so what we want to have at the end is some kind of like a super definition of the gene by aggregating evidence from multiple functional data. Okay, so we just, I won't go into the detail, but I've just code an optimization algorithm that selects the weights that maximize the recall while having a precision greater than 75%. Okay, because I wanted to have like a good balance between high recall, but also like a really, something really precise. So I just fixed the threshold to 75%. Okay. So here are the main results. So in this graph, we have the recall and the precision for around, I think I have like 13 SNP-to-gene strategies. So at one extreme, we have exons that are like perfectly precise, but it's guaranteed through its definition, but has a low recall. Around 10% of the true SNP gene pairs are detected using this strategy. On the other extreme, you have closest TSS, which is not precise at all, but because it links every SNP to a gene, it attains a high recall. So like here, like the recall is equal to the precision. It's around like 35%. And so what we have here is a group of functional assays that that all have a high precision, like a precision between 50% and 80%. But a low recall, right? And so what we observed is that, of course, like this precision and recall is a function of the biosample size of the functional assays, right? So we have EQTL data sets here that use like tens of thousands of experiments. And here we have enhancer maps and single cell attack-seq data that really used few samples, okay? So this kind of makes sense. And this is, I think, is optimistic for this kind of approach, because we kind of thinking like maybe if we increase the number of individual cell types, or if we increase the number of cell types, these SNP-to-gene strategies will improve. Okay. And so now here we have our combined SNP-to-gene strategy that contain 7 strategies, which are all the 7 strategies that I've highlighted here. And so which kind of have like both a high recall, like around like 33% of SNP gene pairs are detected, and a precision of 75%, right? And so this strategy like double either the precision, either the recall of any strategy. So I don't know, at this stage of the project, we are kind of like really happy to what we had in hand. Maybe before I move to the next slide, do you have a question now? Or questions, plural? No? Okay. So I'll take that it was really clear and you are impressed by my results. Okay, so now here are like some features of our new approach. So this approach linked 20% of common SNPs to at least one gene. Nearly all the genes are linked to one common SNPs. We have an average of 1.2 genes linked per common SNP, and around 80 common SNPs linked per gene. One surprising result was what I plotted here, which is like the concordance between all the SNP-to-gene strategies, which shows that there's like a few overlap between all the strategies together. And so it kind of, I don't know. So for me, it was kind of like a strong motivation to continue to combine this map together, right? They all like show different aspects of gene expression. They all have of gene deregulation, sorry, they all have different cell types and tissue. And so maybe there's no one perfect functional assay, but maybe by combining them together, we filling the gap, like the weaknesses of each one has. And at the end, we found like nearly half of the linked SNPs that were not linked to the gene with the closest TSS. So like in half of, a little bit less half of the cases, we have a SNP not linked to the closest one. Okay, so here are more results that I found interesting regarding closest TSS. So here is a precision of the closest TSS stratified by the distance between the causal SNP and the TSS. And so what we can observe is that, okay, the closer we are to a TSS, the more likely the causal variant is to target this gene. Okay, so for example, if a causal GWAS hit has a TSS which is less than 1 kb, then the precision for this, like the probability that the disease variant targets the gene is around 80%. And of course, like you decrease till reaching something close to 0 when you are very far from the closest TSS. And so I don't know, for me, like this was interesting because this was kind of shown for in EQTL, but not in disease, and it kind of also validate our method that I don't know, we have some kind of linear relationship between a distance to the TSS and precision. So here I've plotted the precision of the closest TSS strategy, the second closest TSS, the third closest TSS, and so on till the 20 closest TSS. And we saw, I don't know, something kind of expected, which is like a decrease of the precision. But what was interesting to see at some point is that the precision is not null and converge to a number that is around 4%. And so what does it mean? So our interpretation to that is that from a certain rank, so for example, let's say 15 closest TSS, like we can consider these strategies as linking a causal SNP to a random SNP. And in this null scenario, because if we consider that we have a lot of genes that are associated to disease, then the number that we should have is not 0, but the fraction of genes that are causal for the trait. And so our interpretation of these results is that, I don't know, like the number of causal gene for a trait is around 4%, because this is the precision that is estimated for a random SNP-to-gene strategy. And so I'll comment this number of 4% in further application of my strategy. And the last thing interesting, sorry, is that when you add all this precision by correcting for this type of 4% random thing, then you estimate that around half of the causal SNPs targets a gene which is close by. So basically, it means that if you have a causal hit, you have half of the chance that the target gene is around, so that it's impacting the gene in cis, and 50% of chance that this variant impacts a gene in trans. And I don't know. So for me, like this is some kind of like a huge, I don't know, a huge statement stating that in half of the cases, your target gene is not around. Your target gene is maybe like on a different chromosome. Okay, so I will finish this part by validating our precision metric by comparing this precision metric to two different precision. So on the first graph, I compare the precision estimated using my validation critical gene sets to the one estimated using my training critical gene sets. And so what we observed on the independent SNP-to-gene strategies is that we have a high correlation and a slope not that far from one between the two, which shows that regarding the gene sets, critical gene set we are using, we giving like similar estimate of precision. And here we compare our precision estimates to some kind of real precision that has been estimated using some kind of curated disease SNP gene GWAS pairs. And so the way they did that is they fine map some variants in multiple traits. And for every genes where they had a fine map coding SNP, they assume that all the surrounding non-coding fine map SNPs are targeting this, this gene. And so I don't know, by doing that, we're able to have some kind of gold standard data set. And what we observed here is like, again, like a strong correlation and consistency between our polygenic precision definition and a more classical precision definition. Okay, so now I'll spend the second half of my talk talking about how to leverage this map to either pinpoint a disease gene, either estimate the number of causal genes we have in a disease. Okay, so all this work is some kind of follow-up analysis of this paper, which is a fine mapping method paper that I've contributed to. And basically, like in this paper, we developed some fine mapping algorithm and run it on 50 UK biobank traits. And so we were able to get around like more than 10,000 SNP trait pairs for which we were pretty confident about the causal variants, right? And so then what we did is we just took our SNP gene map and defined some SNP gene disease triplets. And so we are able to nominate 7,000 of them, including more than 3,000 unique genes, which kind of provides some kind of cool resource to know what are the genes that have been highlighted by GWAS and what are the impacted phenotypes. Okay, so now I just want to pinpoint I think two of the most for like interesting example of these SNP gene pair triplets. So the first one is in type 2 diabetes. So what we can see here is like the p-value of every SNP in this region of chromosome 11. So I've highlighted in color two SNPs that have been fine mapped. So these two SNPs are independent, right? They have an R square below than 0.01. What was interesting to observe is that they are not the SNPs with the strongest p-value, and the reason for that is because the SNPs with the strongest p-value is targeting is in LD with these two different SNPs. And so what was really interesting to observe that these two SNPs are both linked to CDKN1C gene, which is not the gene where these two SNPs are. And what was interesting to observe also is that these two SNPs are linked with two different SNP-to-gene strategy to this gene, kind of providing even like stronger evidence than this gene is the accurate target gene and not KCNQ1. And the second thing, what was interesting to observe is that we found these SNPs, so in the EpiMap enhancer dataset, and these EpiMap enhancer were detected in pancreas, which is like a targeted tissue for type 2 diabetes. And so, which means that by, I don't know, aggregating our combined SNP-to-gene strategy and file mapping results, we were able to kind of, I don't know, go from a GWAS hit to a mechanism, because we are able to find two causal SNPs to link them to a genes using either expression and enhancer datasets, and also like validate the cell type or tissue of action. Here we have like another similar example in asthma with two different SNPs. They both again, they are independent of each other, and again, like they both use different SNP-to-gene strategy to target gene BCL6, which is like a good candidate gene, because I don't know, it has some impact on the immune function. And again, what was interesting to observe here is that this SNP was found in an EpiMap enhancer in CD34 plus cells, which is a strong candidate cell type in asthma. So here again, we have been able, like using our framework, to go from GWAS signal to causal variant, target gene, and cell type of action. Do you have any question at this stage?
I did have a question from something a little bit before you got started with this. It was with the correlation matrix, I think, that you had for the different types.
Oh, yeah.
Yeah, I don't know if it's okay to go back to that.
Yeah.
Yeah, I was just wondering, I was trying to go back and forth. It looks like the top half has different values. Am I seeing that correct? Than the bottom?
Yeah. Yes, so it's not correlation. So for example, the way to read that is to say 40, okay, let's pick this number. Okay, so 19% of promoters are validated by GTEx, and 4% of GTEx links are promoters.
I got you. Okay, great. That makes a lot more sense. Perfect. Okay. Yeah, I was a little confused by that. Okay, cool.
Yeah, sorry for that.
No worries. And then I did have a question also. You were talking earlier, I guess, about in a sense, cis versus trans variants, how, you know, 50% of the variants are essentially a cis variant for the next start site. For the other half, like what percentage of those are actually trans in the sense of from a different chromosome, not so much a later start site? Because I think in the graph, it was broke down by second, third, 10th. Yeah. Did you have something showing? Did you have something showing for an actual separate chromosome by chance?
Oh, no.
Okay, cool.
Yeah, no, I don't think it's possible, because how do you pick a gene of a different chromosome?
Yeah, I guess that's the trans EQTLs where you can pick it, but like it shouldn't impact. I guess the start site shouldn't impact that. It might impact the expression through a different manner, in a sense.
Yeah,
right?
I think what would be like super fun to do, we'll take like a raw high-C matrix and just to link every SNP to the TSS with the highest high-C intensity, which is not in the same chromosome.
Yeah. Yeah. Okay. Cool. Yeah. I think
it's doable. I don't know.
Yeah, I think that.
I think it would be fun to do, just to validate.
Yeah, definitely. And then the one other question I did have is how do you see, this is something I've encountered just studying different populations, that there are variants that tend not to be in it, for me at least, GTEx. There are variants because like they might be more specific to like a Asian or African population that don't show up in there. How do you think that impacts, the rest of these results in a sense? Or just, do you have any thoughts on that? That's something I was.
Yeah, I think it impacts. I don't know. I think the literature about like the consistency of gene expression and an answer is really not consistent. Some papers say that gene expression is stable across population. Some say the opposite. Same for an answers. My intuition is that it's different. And so, yeah, I don't know. This is something I could do. Like, I think just taking this map and just, rather than analyzing European datasets, analyzing Biobank Japan datasets.
Yeah, I think that would be
yeah,
cool.
I don't know. I think it's a good master student project. So I don't know if there's like master students in the room who want to work on that. Just tell me.
There's a project.
But yeah, but these were like two good questions. Yeah.
Cool. Thanks. Well, yeah, that was it. Thank you.
Okay. Okay. So now I'll go like to the, I don't know, my favorite part of the talk, which is trying to leverage this combined SNP-to-gene map to try to understand how many genes are causal for a disease. And I kind of feel like it's an important question because I think now the community it's like really open to say that common traits, like human complex traits are very polygenic, which means that it has like thousands of associated variants. But converting this number of associated causal variants to causal genes is, I don't know, I think less straightforward because we have like a smaller number of genes. And I don't know, saying that thousands of SNPs are causal for a trait is not the same thing that saying that thousands of genes are causal, because it will mean that every gene is like nearly every gene is causal for a trait. And so I don't know, like what's the point of doing GWAS if at the end like everything is causal, right? And so this paper, like so this is like a reference from what is called like the Omnigenic paper, suggested a model where you have some of the genes that are really important, and they are called the core genes, and they explain a lot of heritability. And then the less, the further away you are from this core gene in terms of like a network, the less heritability you explain. But at the end, it's really like through like a gene network that you involving all the genes that you're explaining the total heritability. Okay, and so what we can do here now is actually like do this curve, and the way we can do that is by leveraging this fine mapping algorithm outputs, because an output of fine mapping algorithm is like the estimate of the true effect, the true causal effect of each SNP, and then we can link this true effect to its gene using our map, and then by summing this across all causal SNPs, we can have for each gene the fraction of gene heritability it explains, and then we can rank the gene and explain the fraction of heritability group of genes explained. Okay, and so this is what we did here. So we analyzed 16 independent UK variant traits. We did fine mapping on around like 2/3 of the individual. We compute per gene heritability. We rank the genes using this set of individual, and then on an independent set of individual, we computed heritability explained by the top 100, 200, 500, 1000, and so on genes. And so the main result is a red line here. And so what we can see is that we need around 1% of the genes to explain half of their heritability that can be precisely linked to genes. And we need around like 10% of the genes to explain everything. Okay. And so two things we observe next is this, so the first one is that when you use the closest TSS approach rather than the combined SNP-to-gene strategy, you have like a really different gene architecture where like nearly all the genes are involved in a disease, and it's really like the architecture that has been suggested by the Omnigenic paper, but we kind of showing here that it's actually like not the good architecture, like really the good architecture. It's like, it seems that around 1% of the gene explaining half and 10% explaining everything. And so then what we observed is that results from using our validation and training critical gene sets were like pretty consistent, so there was no overfitting. So this means that we are able to go back to the individual estimate of each gene, of hereditary expression by each gene, without considering them as too much biased, right? Okay, and so the first thing we did with these results is to use some kind of polygenic formula, which is from this paper, which give you an estimate of the effective number of causal SNPs in a trait. And so as you can see, like here's the median. So we have like more than 1,000 causal SNPs per GWAS on average. But then we adapted this formula to estimate the effective number of causal genes. And what we observed is that the effective number of causal genes for a trait is around 500, with some number being very low, for example, for hair color, which is around 10, or HDL, which is around like less than 100, to very, very high. So for example, for height and BMI, it's more around 1,000. And for psoriasis, it's something around I think 3,000, which is very high. Okay, then we just ask how like different those genes are impacted by common variant and low frequency variants. So we just try to stratify the signal by the frequency of the SNPs. And why we did that is that because it's well known that because the low frequency variants can have high effects, right? There is less causal variance in the low frequency spectrum than in the common spectrum. Okay, and so what we did is we split this number of effective causal genes into the one explained by common variant and low frequency variants. And what we observed is that we have a ratio of 3 between the number of genes dominating common variant architecture, which was estimated to around 427, and the number of genes dominating the low frequency variant architecture, which was around 150. Okay, and so what was interesting when we had like this number and when we compared these numbers to the one when we mixed common and low frequency variant together, is that, I don't know, like this number is nearly the sum of the two numbers, which suggests like really few overlap between the genes highlighted by common and low frequency variants. And so this is actually what we observed here. So here an example where each point is a gene, and I'm plotting the fraction of common gene heritability this gene is explaining, and the low frequency gene heritability that this gene is explaining. And what you can see is that you have few genes that are in the top 1% in the two architecture. We have a median number of 24 per trait. But really what you observed is that you have some, I don't know, genes that are really specific to some architecture. Right? And like one thing we thought about that, oh, maybe it's just because we kind of missing power. It's just because, I don't know, this number here is just noise because I don't know, like the sample size and like the power to detect effect for low frequency variants is smaller. But what we observed is that this set of common genes and low frequency genes shared the same function. Okay, so for example, here, what I did is, so each point represents a trait, right? And the color represents a set of Mendelian genes for a function related to this trait. And so what we observe is that the top common genes and the top low frequency genes are have around like the same excess overlap in these Mendelian gene sets, showing that, I don't know, maybe these two sets of genes are not the same, but they share the same function. And here we did the same by looking at overlap across genes that are cell type specific. And so, for example, what we can see like is, I don't know, if we're looking at MIT platelet volume as a trait, we observed that the excess overlap for the low frequency genes and the common genes is the same and is like very high, like for genes related to blood and immunity. Which kind of, I don't know, kind of for me, like it was kind of a striking result. I just think, okay, like based on the frequency of a variant, like the genes targeted won't be the same, but like the function will be. I don't know if it's clear for everyone, but I have five minutes. I think it was my last slide of results. Yeah. So I know I'm going to conclude on that. So so what I've shown you is that we've developed a framework to evaluate and combine SNP-to-gene strategies using a polygenic approach, and that I don't know. And I don't know, like kind of concerns the fact that we don't have like a GWAS SNP gene gold standard dataset yet. When we applied this framework, we observed a lot of functional SNP-to-gene strategy that have a high precision but a low recall, and so a way to aggregate this information was just to optimize their combination. And so we've developed this combined SNP-to-gene strategies that maximize recall while having high precision. And so when we used this map to analyze GWAS data, we're able to highlight a lot of SNP gene disease triplets with sometimes interpretable function. And then we observed that around once linked like this polygenic signal to the genes, that around 1% of the genes were able to explain nearly half of the heritability precisely linked to all genes. And that we observed that these genes were impacted like differently by common and low frequency variant, but we are like keeping the same same function. Okay, so I would like to finish this talk by acknowledging all my collaborators on this project and just saying that, I don't know, I have a new lab at USC, and so I'm looking for motivated graduate and undergraduate students and postdocs. And I'll stop here. So do you have any questions?
I have a question, Stephen. This is Keith Burkhardt from USCISI. You're saying that low frequency genes do not have much overlap but have common functions. I'm not super familiar with genetics. I come from the field of computer science. So to better understand what you mean, by low frequency genes, do you mean common versus uncommon SNPs, or what do you mean precisely by that?
Sorry. So common variants are like what we like, I don't know, what we are familiar to analyze with GWAS. So they are like variants with a frequency greater than 5% in the population. But with the traits I've analyzed, which are UK Biobank is so big, like it has like nearly 500,000 individuals. So we are powered enough to look at variants with a frequency between 0.5% and 5%. So I call them like a low frequency.
I see. So basically, it's the common SNPs versus the very or the SNPs that have that are more equal in their distribution versus ones that are very unequal, right?
Yeah.
Yeah, I see.
And so what I've shown is that when you have like common hits and low frequency SNPs and low frequency hits, sorry, in a few scenarios, they target the same gene.
I see, thank you. The other question I have is that, well, from what I understand, you're basically showing that while your method has high precision, it doesn't target many, it doesn't target all SNPs. I mean, you're inevitably limited by the strategy. So in many scenarios where we might not have, where we might not be using the SNPs that you're using to transfer them into causal, to figure out what genes are associated with those SNPs, do we have to just rely on TSS in general?
No, I don't think so. So I kind of feel like really like the signal is split in two, like what you can explain in cis and what you can explain in trans. And I kind of feel like when you can explain this in cis, maybe it's very likely that you will have like functional data that go into this direction. But if you have like no SNP-to-gene strategies, maybe it means that the impact of the SNP is in trans. So I think I would prefer like looking at nothing, like and forgetting the SNP rather than assuming that it's the closest one.
Okay, thank you.
Other questions for Stephen?
Yeah.
Well, if not, thank you for coming back again. Looking forward to hearing you. I mean, I enjoyed hearing you.
Thanks.
So thanks all.
Thanks.
Bye-bye.
Okay. Have a nice day.
Bye.